started. So the next speaker is Professor Aleksei Aksimentiev from University of Illinois at Urbana-Champaign. And as you all heard, that he would also like to take questions in the middle of his talk. So if you have any questions, just you can raise your hand or type in the chat box whatever you are comfortable about. So he will talk about DNA in tight spaces from nucleosome and chromosomes to origami and viruses. Professor Aleksei.
Okay, thanks so much for inviting me to give this talk over Zoom. Okay, I hope everyone is feeling fine and that you can hear me well. Never know if the mic is strong enough. So I have actually lots of slides. So I was planning to speak for an hour. So if you want me to speak for less, please let me know. If not, I'll do that. So, and yeah, please feel free to interrupt me as I go. All right, so before I go to the DNA subject, let me kind of tell you a few other things that we do here. So my lab at University of Illinois, we are strong in nanopore sequencing. So we model all kind of nanopores for protein and DNA sequencing and RNA sequencing and so on. We also work on biological condensates. I noticed that that subject was covered in the program already. I'm not going to talk about it, but we have a big effort in that area. We are interested in nuclear port transport, have like one of the largest simulations that we've done to date, like 160 million atoms. Not going to talk about it. And we also have a very successful program in designing artificial membrane channels, which again, I'm not going to cover. But if you're interested, you can ask me a question or you can look at the publications at my group's website. So I'm going to tell you about DNA and our efforts to understand how it functions in tight confinements. And I'm going to use the following instrument here. It's called all-atom molecular dynamics, the computational microscope. So that's something that I learned from my mentor, Klaus Schulten. So he always said, use a computational microscope to look at the molecules. And that's basically what we've been doing. So what it is, it's a really big computer. You're looking at the picture of blue waters that used to be one of the bestest and fastest computers in the world. Now I think it's not even top 100 because the computers, they age. But anyway, it's still a very impressive computer system. And with that computer, we can animate in time microscopic models of biomolecules. So for example, you're looking at a DNA molecule going through a nanopore. It should be a movie. If you don't see a movie, please let me know. So what can we do with this method? We can image systems that range in size from single atoms to billions of atoms, and the processes that we can image with this instrument are typically range from nanoseconds to hundreds of microseconds. All right, so let me kind of start with the beginning. This is how I got into the DNA business. So as most of you know, same charges repel in vacuum, but when you put them in a medium, you can get pretty much anything you want depending on the medium. So same charges can attract, and there are many examples of that. One, I guess, interesting example for physics oriented people are the electron pairs, that form to enable superconductivity. But here we're talking about DNA. So here's the DNA molecule, and as you probably know, DNA is in water. It's surrounded in electrolyte. So it's not just the DNA alone. DNA is negatively charged, so it comes with ion atmosphere. And experimentally, it has been established that the interaction between two DNA molecules depend on the
Excuse
type
me,
of
are you still on your first slide? Or
Yes, your slides are not changing.
Fantastic.
Now we
Great.
can see the fourth slide, I think.
Great. I was great. Yeah, by the way, if you are if you type something in chat, I'm not gonna see it. I cannot read chat and give a talk at the same time. You actually have to say something.
Maybe
So,
you
all
can
right.
make it full screen, Alec.
Yeah,
It's
we'll
not
see.
full. Yeah. Oops.
Let's try again. This sometimes happens. I could never figure out why. Why sometimes it works, sometimes it doesn't. Let's try again.
Yeah, now it works.
Yeah, I mean that will definitely work. The question is if you're gonna see the next slide. That's the question. Okay, but basically I already told you about this. And yeah, the movie was fantastic, but I'll show you more. So the point is, depending on the charge of the electrolyte, you can have repulsion or attraction. And that was kind of the first topic that we wanted to study was all atom molecular dynamics. Okay. Now it should be a different slide, is it?
Yeah, now it's different slide. Fine.
Great. All right. So here's how we do, how we set up our molecular dynamics simulations. I know some of you are experts, but let me just show how we do. So we take a molecule of DNA, and but well, we have a big computer. Let's take more than one. This is 64 molecules of DNA. And in this particular case, we make them effectively infinite. So we add cations. So this one is a polyamine. Each has a charge of +4. Then we also add some background electrolytes. So this is NaCl. And then we add water. So this is really like an experimental system, if you think about it. We have everything that experiment has. And to characterize the interactions between DNA molecules, what we do, we confine this DNA bundle to a potential. And that potential will make sure that the bundle stays together, okay? So think about it as a kind of a force that acts only on DNA and allowing water and ions to pass through the potential. And then the rest is described by conventional molecular dynamics. I'm not going to introduce it, but the point is that there are parts that are responsible for bonded interactions that are set by quantum mechanics. And there's also part that is non-bounded, where there are charges, and then there's the van der Waals potentials. And the van der Waals potential is kind of always a little bit ad hoc. Okay, so that's the system, and here's an outcome of one of the simulations that we've done. So what you're looking at, and again, this should be a new slide, you're looking at a top view of the DNA array. So each of the circle is one DNA molecule, and you see there's this little dash there. So the dash there is to indicate where the major group of the DNA is. All right, so so here's the outcome of the simulation. The simulation runs, the DNA moves, and in the background, you're looking at the local concentration of ions, right? And well, I mean, it runs, so it was good. The problem is that you can clearly see that it kind of forms clusters, right? And experimentally, we know it should be hexagonal. Now, if you also look at the spaces between DNA, you'll see that there's very high concentration of ions there. And if we examine what actually happens there in details, we'll see that those ions would facilitate those bridging interactions between DNA phosphates. And then, well, we set it up, our system, so you can do one-to-one comparison with experiment, and that's what we do. So I hope you can see my cursor. So what you're looking here, this is experiment, raw precision, the golden standard. And this is the result of our simulations, these different molecular models. And this is logarithmic scale, okay? So we are nowhere near what it should be. And that was kind of a big disappointment. Basically, if we do it for magnesium electrolytes, that's just totally off. If we do it for spearmint, which was this polyamines that I showed you at the beginning, we are getting basically qualitatively wrong results. Okay, so that was a big disappointment, and we thought that maybe there's something wrong with the experiment. Of course, that's the first thing that you think, but no. Of course, it's a golden standard experiments. It's right on. So the problem was with the parameters, and we fixed it. And the way we do it, I mean, when I say we, it's actually Jijun Yu. So he did most of this parameterization. So what he did was to use so-called osmotic pressure calibration of the force field. So the idea is the following. So instead of looking at DNA, complex system with lots of parameters, he chose to look at interactions that matter. So for example, he looked at the sodium ion and this chemical substitute of the DNA backbone. So this is dimethyl phosphate. So you have a phosphate to metal group. Actually, the oxygen here is the same atom type as of acetate. So you can also use acetate for it. And then, so here's a system, and it has two compartments. One is where the ions are, and the other one where the water is. And there's, again, the semi-permeable membranes that we can measure how much pressure we need to apply to maintain the solution at a given concentration. So it's a direct readout of osmotic pressure. And voila, simulation runs. And so most importantly, we can get directly the osmotic pressure from the simulation.
There is a question. There is a question for you. It says that what does the pressure signify here? Is it the osmotic pressure of the DNA? I think you just answered that. This is Shupriyo asking the question.
Right, exactly. So in this particular case, it's osmotic pressure of the electrolyte solution. In the previous case, it was osmotic pressure of the DNA bundle. Yes. But so what we found from using standard force fields, that osmotic pressure in a DNA bundle was too low. That's why we went through this effort to refine the force field. And so we are using those model compounds, and that's what we find. So the plot here shows, the standard osmotic pressure. And this is experiment, so you can see they're really different. And the way we fixed it, we changed the van der Waals parameter. Well, actually, what happens if you simulate it, you just get a cluster of ions. I don't know if you ever simulated sodium chloride at 3 molar or 5 molar, you'll get a cluster. And it should not be a cluster. It should be a dispersed solution, because everybody knows what solubility is. But that's what you get with standard force field. So what we did, we changed the van der Waals radius using so-called NB-fix correction, just by a tiny bit. It's a fraction of an angstrom, and that kind of solved all of our problems. And we did it for lots of other combinations of ions, and produced what we call CU-fix, which stands for Champaign-Urbana fix for amber and charm force fields, which you can get from our website. But long story short, it worked. And now we can finally look at the physics of DNA systems with this refined force field that produce that can be used to make predictions about systems. So how does it work? You're looking at, again, a system top down. This one has polyamines and the initial conformation of the DNA helices. And so the simulation runs, everything moves around. I hope you can see the movie. And so what do we get here? Well, first of all, the distribution of DNA is much more rational. So it's a hexagonal pattern. Ions explore their conformations. And we actually get the right pressure. So if you look here at the graph to your right, there's a curve for sodium, and this is what we get. It's right on. There's a curve for spearmint, and this is what we get right on. And again, I emphasize we did not use DNA simulations to fix it. We used the model compound simulation to fix it. We used DNA to verify it. So in a way, it's a verifiable prediction here. And so good, we are very happy with this outcome. And then, well, what kind of physics can we do with it? So first thing was to test, which of the models of DNA condensation actually happens. So there are two popular one. One is like a Wigner crystal model where DNA where polycations bind to the DNA, and then somehow its matching pattern happens. And the other one is the bridging model, and it is the bridging model. And how do we know? Well, we can actually make a Wigner crystal scenario where we artificially bind pyrimidine molecules to DNA, and then basically the osmotic pressure gets the same as with monovalent salt. All right, what else did we learn from these simulations? And it's all described in this paper that is listed here, which is also one of the things that I suggested to post. So first of all, the DNA orientation is correlated. So if you look here at 3 different orientations, it's maybe not very clear, but here you have minor groove to major groove. It's shifted, and this is major groove to major groove. There's actually a preference for DNA being oriented 90 degrees with one another. So you can kind of see it here as a peaks in the orientations. So the DNA forces are pairwise. That was a little bit disappointing. We were hoping to find some interesting physics there, but basically, if you know the force between 2 DNA molecules, you can tell what the pressure in the array is. And we know it because we do both the force in the DNA between 2 DNA and in the array. And we also looked at the hydration force, what is its contribution to the attractive force between DNA molecules. So that was actually not a trivial thing to do, to compute the forces from molecules only, from water molecules only. But what we found is that the water usually, I mean, most of the time, always has a repulsive term, so it doesn't actually produce any attraction. It's the ions that do it. And finally, we can also characterize things like friction between DNA molecules, how it depends on the distance. So there's like really a bunch of interesting stuff, but that's published. So let me move on to another subject, and that one has to do with how DNA is organized in biological cells. Most of you probably know that there are different levels of DNA organization, starting from double helix to nucleosomes, and then through several scales, we end up with something in the nucleus at the scale of 10 micrometers. And there, of course, it's clear. We know that it's not random there, right? We know from experiments, so-called High-C experiments, that the structure of the DNA at the chromosome level has an order. And how do we know? Well, I'm not going to explain in details how it works, but basically there is a way to correlate which sequences are proximal in the nucleus. And the pictures that you get from these experiments are the following. So you have a DNA molecules, and they are like 80 ridge domains, and they typically form closed structures. And then there are also CG ridge domains that are more loose. So by 80, I mean 80 nucleotides and CG nucleotides. And experiments also suggest, that that there's also similar organization between different chromosomes. So, have different pieces of DNA there. And yet, they also show this propensity to colocalize. The 80 molecules kind of tend to be together, 80 rich molecules, whereas the GDC rich molecules are not. So that was interesting. Of course, there are many biological reasons for that, but we were really kind of inspired by work of Kornyshev and co-workers that suggested that maybe, it can come about just because of the physics of DNA. So here's how we probe the interactions between 2 DNA molecules. It's a fairly standard method. We have 2 molecules in electrolyte solution, so I'm not showing you water, showing only 2 molecules and amines. We have a harmonic spring between them, and that spring is used to measure the effective force. So if we do the simulations for long enough, we'll get something like that, potential mean force. I mean, well, we get the force versus distance directly, which we can integrate, and we get the potential mean force. So in other words, we get a free energy for bringing two DNA molecules from infinity into close contact. So what you'll find here is that, well, first of all, it's negative when two molecules are close. That's good. That means they actually attract. And that simulation was done using a CG repeat DNA. And when we did the simulation using the 80 repeat DNA, that was a little bit shocking, but we got a different curve. You see, it's shifted down by about 1 kilocalorie per mole per turn. You might think it's not a small number, but it's definitely there, and it's per turn. So if your DNA has 1 million turns, that's a lot of KTs. All right, so that was shocking, and but also interesting, because that's kind of similar to what you see in biological systems. Why does it happen? And that's the power of the computational microscope, because what we can do, we can go now and look at our trajectory and ask the question, why do we see what we see? And the reason is that, you know, if you look at what happens between DNA molecules, you'll find that for 80 systems, you have lots more of those polyamine in between of the helices. So that's so-called bridging polyamines. And you have some for CGs, but there are fewer of them. And the reason for that is actually chemistry. So we were also looking, you know, being in the physics department, always look for some cool physics, but it was chemistry. So the reason why those molecules are between helices is because they actually physically cannot bind to the grooves of the DNA that has a T nucleotide. So you see, the T nucleotide has this methyl group, and it's like a little steric bump there that prevents polyamines to get lodged in the helix. And that's why it happens, right? So that's the mechanism, and then Ji Jung thought, well, okay, good. What else has a methyl group? And of course, the methylated DNA has a methyl group, as the name suggests. So you're probably familiar with epigenetics. So basically, you have a DNA code, but then there is a way to modify it. And the most popular modification is so-called methylation of C. So adding a CH3 group to the C nucleotides. And that really changes how genomes is transcribed in different cell types. You know, if you think about it, we all our cells have the same genome, but you know, your hair cell looks different than your liver cell and so on. And that's because different things are on and off, and this methylation kind of tells the machinery what's on and what's off. So in our particular case, we just look at a very simple system. So if you have our piece of CG repeat DNA and the free energy, what happens if we methylate it? So if we methylate it, well, then we get a different free energy. You see, just like we predicted, you know, we add methyl group. So methyl group will make the DNA attract more strongly. And that's basically what our prediction was. And it could have been an interesting, well, by the way, this actually agrees with what we know from experiment. What happens with the chromosomes when they get methylated, you know, they become more compact. And that would have been just an interesting simulation study if it wasn't for the fact that Ji Jung was a friend with Hajin Kim, was a postdoc of TJ Ha, who is a world expert in single molecule FRET experiments. And at some point, they had lunch, and the expectation was that the experiment will prove that simulations are wrong. So here's an experiment. It's super clever and amazingly done. So you have a substrate to which you can bind a lipid vesicle. And there is a technology to make sure that that lipid vesicle has only 2 DNA molecules on average. And you can measure one DNA molecule, you can label one DNA molecule with one fluorophore dye and the other one with another dye. And when they come together, you get what's called fret. You get a signal, and when they're apart, you don't get a signal. And you can just measure how frequently those molecules come and use that as an indication of attraction. So that's exactly what Hajin did. And if you look at the one at the, if you look at the graph at the left, so you see the graph that shows how frequently DNA molecules carrying CG-rich sequence come together. So they do come together once in a while. But then if we just change the content of the AT, so we change the sequence so it has higher AT content, we get much stronger fret. We can also do it with the CG sequences. So we can methylate them using enzymes, and then we again get a much stronger fret, meaning that they attract stronger. So that was a kind of a direct proof of the mechanism that we proposed. And that was really satisfying. It's one of those things where we could predict something using all-atom simulations. So here's a picture that emerges from those studies. So really, when you have 2 DNA molecules and the electrolyte that promotes condensation, the strength of the interactions will be modulated by the DNA sequence, and the strongest condensation will be for AT-rich sequences, followed by methylated CG, and the least condensing one are the CG nucleotides. So that was nice. So after that, we did another study where we looked at the mechanism of condensation produced by lysine molecules. And the reason why we did it is the following. As you may know, in biological cells, in eukaryotic cells, DNA is wrapped around histones. And those histones, they carry so-called histone tails. So those tails are rich in lysine residues, and they are there, they're there to modulate the interactions between DNA. And so what those experiments showed basically that everything that we found for spearmine actually holds. So for example, in this particular experiment, you're looking at fret efficiency versus concentration of lysine tetramers. And so basically experiment like that, you add more and more of this lysine solution, and you're looking at when the fret efficiency starts to rise. So if you look at this curve, so the first one to rise, meaning indicates the stronger attraction. So the first one would be the AT-rich molecules. Then you would have a methylated CG molecule, then you would have a CG. And this one is very interesting. This one is a DNA construct that has a U, the RNA nucleotide. The RNA base, it's still the DNA molecule, but has an RNA base, and it has a least propensity for aggregation, which was actually very cool because that has been previously shown that RNA actually resists condensation. And if you look at the chemistry of this U molecule, it will become obvious. U doesn't have a metal group. It's an equivalent of T, but it doesn't have a metal group. That's why. There's fewer bridging ions, and that's how it works. So having established this sequence-dependent forces, of course, the next step is to say, well, okay, what do they do? Well, what can they do in biology? And being trained in phase separation is kind of obvious that they would phase separate, right? So if you have a mixture of molecules that carry AT and CG sequences, if you mix them together, they will undergo phase separation, just like illustrated in this movie. And so our big picture message here from this work is that if you look at the long DNA molecules that has a heterogeneous sequence and you put it under conditions where the DNA condensation is possible, it will microphase separate into domains that will be AT-rich, forming condensing phase and condensed phase, and CG-rich domains that will not be condensed. And that is actually consistent with what you see in biological systems. But of course, the question still remains how much of it can be transferred to biological system because they're so complex. All right, so that was this part. Now I'm gonna tell you a little bit about DNA flexibility, and this work is done in collaboration with TAGGIP. So the idea here is, and that's of course not new, but you probably know that you know, the DNA, in addition to carrying the nucleotide code, perhaps it has a secondary code, and that's the idea that goes back to Jonathan Widom, that determines where nucleosomes are on the DNA. And so why is it important? Oh, okay, so here it is. So, you know, the DNA is wrapped around nucleosomes, not all of it, but some of it. But then, let's say you want to transcribe a gene. Yeah, if you want to transcribe a gene, an RNA polymerase should be able to come and bind to it, and it will do it only if you have a loose fragment there. You know, it will bind, and then it will go on this ride and start making a messenger RNA. But you know, if the DNA is tightly bound, well, the reading cannot start. So that kind of provides a very nice intuitive mechanism for controlling which genes are on, like which parts can be transcribed, which parts cannot be transcribed, by modulating the mechanical properties of DNA. And again, this idea has been around for a while. So what experimentally how it can be probed, and this is one of the ways, is so-called DNA looping assays. And that's something that was pioneered at the single molecule level by TAGGIP and his lab. So what you have here is a single molecule of DNA, and it's labeled with two fluorophore dyes. And eventually, you know, it will form a circle, a loop like this. So here's the animation. Boom, done. And you or others can do this kind of measurements, looking for the time it takes for DNA molecules to form those loops. So that's so-called DNA looping assay. It's kind of conceptually very simple to understand, but the results were really striking because, so if you look at the looping time, which is typically in minutes, you'll find a really dramatic difference in the time on the DNA sequence. So you change the sequence by a little bit, and it gives you really different looping times. And that basically was a puzzling observation, and also a direct measurement of how stiff or how flexible the DNAs are. So we were interested in looking at this from a computational point of view. Of course, this is really kind of a difficult problem if you think about it, because those looping times are, I mean, it's hours. So we look at it from a different approach. We said, okay, well, since we cannot really simulate looping in a LATAM MD, well, let's just look at the loop itself after it's formed. So here's our typical simulation system. We have a nicely formed loop in a box of water, and let's say we run. So of course we have water ions, everything is in there, and okay, so here it is. So this particular case, you're looking at the plot of the two distances, and the point of this plot is to say that it actually stays looped. Okay, so there are some previous studies that found that if you make a loop like that and you run it in molecular dynamics, it will form kinks and so on. So in our hands, it doesn't form kinks, and the reason is that we do very careful equilibration. Basically, what we do, we enforce base pairing for the first 100 nanoseconds, which is enough for the DNA to go this inside-out transition, and then we let go. You know, and then we can simulate. This is actually kind of a little bit outdated slides. I think the simulation now is 15 microseconds, and it stays as a loop, you know.
Okay, there is a question.
Sure.
There is a question from Abhishek Agarwal. He is asking, is there any restraint applied during the loop simulations?
Oh, that's a great question. So to initiate the loop, yes. So what is there is, so we build a loop, and it's, I mean, it's They're not nicks. They are harmonic restraints between two atoms of each base pair for the first 100 nanoseconds. Okay, and that was very important because within this first 100 nanoseconds, the DNA goes inside-out transition to find its minimum. After that, we remove the restraints, and it's completely unrestrained simulation. It stays as a loop for 10 microseconds. Yeah, so that's like simplified picture. And all right, let's have a look. So you already saw that it goes this inside-out transition. I was going to bring a piece of of a rubber band to show you. Unfortunately I forgot. So basically, it's like, I don't know if you can see me, but if you have a piece of wire and you will try to move it inside out. So that's the kind of transitions that we're talking about. And that's what illustrates here. So, that's called a register angle or poloidal angle. I think that's a mathematical definition. It's like inside out transition. So for this particular case, so look, there is this one atom here, look at it, what happens. So it kind of stays inside most of the time. Sometimes it goes away, sometimes it comes back. But it undergoes, the DNA undergoes a spontaneous inside-out transition. And what was interesting is that there's definitely a preferred angle for it. There are fluctuations, but it's set. And then if we start with a different initial conformation, the DNA quickly I mean like hundreds of nanoseconds, would find that equilibrium poloidal angle and stay there. We can do it many times. So we start from all kind of initial angles. It all converges to the same one if the DNA loop has the same DNA sequence. Now, we did it for 40 different sequences, and we actually find that the sequence actually tells you what the angle is. So in other words, the DNA sequence tells you what part of the DNA circle will be inside and which part of the DNA circle will be outside, and it's fairly robust feature. So that was our discovery, and we tried to understand it quantitatively by doing the following. So what we could do, we can compute the free energy for this inside-out transition, and if you're interested, we can ask me how we did it, but basically we measured the torque required to maintain a certain poloidal angle, and then we can integrate the torque, and basically we get the free energy for this inside-out transition. So that's what we have here. So don't worry about this quote, the scale of the axis. The point here is that there is this minimum, which is basically where all those trajectories go. And then there is this kind of a metastable state. So what's the message here? Well, first of all, there's this free energy for inside-out transition in DNA circle, which is right on with what we observe in free equilibration simulation. And why is it there? So that's a good question. So if you look at the DNA circle carefully, and again, I hope you can see my cursor here, it's actually not a circle. It's actually an octagon. So you see there's a straight line here, and there's a sharp band, straight line, sharp band, straight line, sharp band, and so on. So here in this circle, we can plot local deformation of the basis, and you see it's deformed basically where it is deformed where major groove of the DNA faces inward. It kind of makes sense because you have space there, so it deforms where you have major groove. And now if we're going to turn it inside out, so here's the movie, see, we're going to turn it inside out. This deformation, it persists where the major groove faces inward, because that's the only way it can actually, that's how the geometry of the DNA works. Now with that in mind, and then of course the DNA doesn't want to deform. So the equilibrium angle is really, you think about it, it's like minimum frustration in the energy, and we can describe it as a quadratic function of local DNA bending, and that's what we did. So lucky enough, we had blue water, so we could do it for 40 different sequences. So we computed this free energy, kind of a monumental simulation. It was a lot of computer time went into this. But at the end of the day, we obtain, this kind of a new parameterization of DNA stiffness at the level of dinucleotide approximations, where, for all combinations of DNA nucleotides, we have a stiffness parameter, including the methylated DNA. I think that's something that has not been done before. And good, so we've done it. Now, did we learn anything from it? And again, this paper should be published at the beginning of the year. I'll just give you a glimpse of what can be done with it. And again, this work done was by Ji Jung. He's now a professor in Korea. So what we can do with this energy function, now we can go and do, yeah?
There is a quick question from Nihal Mathur. What is S? I think about your previous slide. What is S? She's asking about some notation. What is S?
Yeah, so S is the index along the DNA. So if we have 90 nucleotides, 19 base pairs, so they will be numbered from 1 to 90. So then in this function, so the energy is a function over dinucleotide steps. So the steps are from 1 to 90 because there are 90 base pairs in the DNA. And there's a local deformation, and there's an energy function. So the local deformation we kind of already know, because for a given shape we can tell what the energy is, and even the nucleotide sequence. So hopefully that's clear. Is there another question about this?
No, but I have a question actually. Maybe it's a very stupid question. You are showing energy costs due to bending or twisting, whatever you call it. But is it not possible that the loop actually also stretches? Or is the contour length, the total contour length, that always remains conserved, is it?
Yeah, that's a great question. It is in our hands. We don't see any melting of the DNA. The local distance, the structure is actually well preserved. So no, it doesn't. Of course, the circle doesn't remain circle globally. If you look at it, there is a little bit ellipticity and there's some fluctuations there, but most of the time the length stays the same.
Okay, thank you.
Sure. All right, so after we've done all of this, what's the point? And the point is that now we have this function and we know how DNA is bent in the nucleosome, and we can ask questions like at the genome scale. And, basically we can connect physics to omics. So give you an idea what it is. So here in the black curve, this is experimental nucleosome occupancy over a 20,000 fragment of chromosome 1 of yeast. And that's the black curve. And the red curve is what we predict, what Ji Jung predict the nucleosome occupancy should be based on our parameterization of the model. So you see the correlation is really striking. Another example here is you can look at all of the transcription start sites. When you have a DNA, a certain part of it is designated where you start transcribing. And again, the nucleosome positioning would have a local minimum right before the transcription site. And it kind of makes sense because you don't want to have a nucleosome where you want to start transcribing because things cannot bind. So you have this lower nucleosome occupancy, and we could have basically predicted it from our model. And so on. So there's lots of stuff here, including how methylation affects it, and when it's published, I encourage you to read about it. So I am running late here, so I'm going to switch gears a little bit and try to move faster to DNA origami. So by the way, I have 15 more minutes, right? I just want to make sure I don't hold anyone. Okay, good. Yeah. So in the 15 more minutes, I will kind of give you an idea of what we do in the space of DNA origami, and then I'll show you a little teaser about viruses. So with origamis, which you probably are all familiar, so the idea is that we can make anything out of DNA, and that's something that Paul Rosamond came up with. I mean, the specific ones that I'm going to show. You have a viral genome, you add staple strands, and you add enough of them so that every part of the DNA is covered. And then experimentally, you would heat it up, and then you cool it down, and it will self-assemble into whatever you program it to be. So in this case, it's of course logo of the University of Illinois. But people have done all kind of things, starting from smiley faces to membrane channels to this a device to measure forces. And so those are like renderings, what the people want it to be, and those are the actual images of what people get. So it works very well. It's amazing technology that is becoming super popular. All right, so we were interested in modeling those systems. If you ask why did we do it? I think the first one we did because it was there, but then of course we had some scientific questions to ask. But really, it's interesting, because those DNA are really confined, because they are there, they're confined into space not because of counter ions or pressure, but because of the crossovers in the design. So there are several ways to look at those structures. The most popular one, of course, is this transmission electron microscopy. So you just take images of stained DNA. And there are several computational methods that existed before us, like CanDo is a really good program to look at structural fluctuations in the DNA assemblies. And but unfortunately, they don't crystallize. Those systems, which is a little bit ironic because they were developed to facilitate protein crystallization, but they actually don't crystallize. And the only way to look at them, like actual structure experimentally, was this Cryo-EM reconstruction. So we thought, well, okay, well, maybe we can replace Cryo-EM with our all-atom electrodynamics. That was very ambitious. So let's see how they compare. One expensive tool against even more expensive tool. Let's see. So this is kind of a data that you would get from cryo microscopy. You look at so-called pointer structure. So it's an object that was designed to be asymmetric, so you can align images of it to produce a cryo density. So here's the density experimentally derived, which you can use to build an all-atom structure. So this all-atom structure derives from cryo-EM microscopy. So it's experimental structure, but you can also do it in a simulation. So it was, it was a big simulation. At that time, 7 million atoms, everything 1-to-1, and we just ran. And to our delight, it actually starts to deforming towards what the experimental system is. Just like that. And if you look at from the other side, you can also see, I mean, at first it looks like the deformation are a little bit minor. They're actually not so minor because they're very deliberate, and it definitely goes into the right conformation. So here's two densities. One is experimental, the other one is cryo-EM derived. I don't know which one is which. The caption will tell. Yeah, so on the right is the all-atom MD, and we can kind of do this comparison more quantitatively. So there are two models overlaps, and one is all-atom model, the other one is simulation. So you can see we do really good job there. Not just the global structure, but also those little funny features. I mean, one thing you can see is that we have more atoms in simulations than in experimental structure. That's because the parts at the end cannot be resolved. But all those holiday junctions, playing of the helices, also at the end of the structure, we could reproduce directly. So this is a stack of holiday junctions that was made there by Henrik Dietz on purpose to see what happens. So we got the geometry there also right on. So basically, the conclusion was that our all-atom model using you see there's a little bit out of plane motion and also splaying at the end. That all came out just from the simulation, meaning that our model is actually pretty good. The problem was that that was a hero simulation that only we could do, and it's definitely not something you would use to design structures because no one has a petascale system in their back pocket. So that's how we come up with this method called energy MD. So what does it stands for is elastic network restraint guided molecular dynamic simulations. So basically, it's a kind of an implicit solvent version of a molecular dynamic simulations where we use our knowledge of how DNA deforms to predict the structure. So it works pretty nicely. If you want to try, we have a web server that you just upload your design and you get everything to run the simulation. We don't run it for you because it still takes a day to run it. So that was nice, but actually the ensemble of spaces is much more diverse in DNA origami and similar self-assembled structures, you know. So if you look at like this DNA vases or gears or buckyballs, you know, those are huge things, and we would like to have a tool to be able to predict their structure and examine properties. So here was the key idea, and this work by Chris Maffio, a postdoc in my lab. So the idea was that, well, we don't actually have to look at the structure at the same resolution all the time. So let's just do this, you know, let's just refine resolution as we go through it. So that was the idea. So the resulting model called a multi-resolution DNA model, or Mr. DNA, which is Chris. So this is how it works. Just give you an idea, looking at the fragment of DNA simulated at different resolutions. And the nice thing about it, the DNA has the same properties. At any resolutions, it's the same persistent length, same everything. And the non-bounded interactions, so we did a good job there. Basically, we used, again, this Rao-Parseghian model. So we get the same pressure in DNA array regardless of the resolution, whether it's, you know, 1, 3, 5, 10 beats per fragment. And we also made sure that they kind of, you know, cannot cross. So that was actually important to make sure that they do not cross, because usually with many coarse-grained models, you would have like crossing between DNA molecules. And okay, so it actually works. So we have an automatic workflow that you would start with a design, and then you would have a kind of a straight helix. Will be relaxation at high resolution. Then we would refine it to a lower resolution, and even higher resolution and even higher resolution, and then we would end up with a Latin model. So this whole thing typically takes 30 minutes, and it's automatic process. So here's an example. Again, this is the pointer structure. We simulated at high resolution, then we increase the resolution, and at the end of the day, we end up with an all-atom model. And it's basically as good as our hero simulation using blue waters, but you know, someone has to do it. So we did it, but now we know we can use the result to learn something about, and voila, here's the model. So it can be used as a structure prediction tool. So now there are quite a few Cryo-EM reconstructions from Henrik Dias' lab, and we are able to reproduce most of them. You know, so the only thing is you have a design and you know that it falls, but then we can tell you three-dimensional structure with very high accuracy. It's actually the same accuracy as the reconstruction of the Cryo-EM. So here are examples of the band structures. So those have a programmed band, and we just do the band right on. The program comes with a flexible interface that you can assemble structures that are difficult to assemble otherwise. For example, this DNA box. And again, in the illustration, you know, we start with a coarse model, and then we refine it to more like finer detail coarse-grained model, and then we can do the all-atom structure. So we can also do things like Flask. So this is a really difficult system, because if you take the Cardano design, you get a sheet. But because we have this Python interface for it, we can transform it into a Flask, and this is how it is. I mean, it is a simulation, so the resulting structure is not symmetric, and it's actually not symmetric in experiments. There's this funny dip, develops here, and if you actually look at the experimental picture, sometimes you can find it there. We are not limited to origami, so we can do all kinds of structures. So the tessellated structure, this one is from Bjorn Holberg, the bunny. He made the cover of Nature, really impressive work. Basically, you can take any shape and cast it in terms of DNA. And so what our contribution to this is now we can take the design and kind of make it live. It's our system is very easy to use. So this is something that Sartak did when he was a rotation student. So I'm very happy that he joined my lab. But when he was a rotation student, that was his project, to simulate this nano gears, like the one on the left, and this Bennett linker. So those are really useful things because it's a coarse grain simulation, but you can explore the degree of structural motion. And in the case of Bennett linker, we can actually compute the angles and compare it to Cryo-EM topography reconstruction, and it works pretty nicely. So this has been published. It's on our website if you're interested in DNA origami simulations. I think that's a really good start. We have a tutorial and everything, and we also do export to OxDNA if you're interested in using OxDNA for it. We'll give you a good starting point for it, and then of course from OxDNA you can also go to OLATOM and all of that. All right, so last 3 minutes, I'm gonna give you like a teaser of what we do with viruses. And viruses is another system where DNA is highly packaged. Like there's lots of DNA there. It's a highly packaged system. And there are lots of questions about what is the structure of the genome inside viruses. And I think we know the answer, but I'm gonna just give you a hint of what it is. So here's how we did it. We took a structure, it's this Cryo-EM reconstruction of a HK97 virus. So we use this specific virus because a lot of it is known. So it's not deadly or anything. It affects bacteria, but there's a lot of information about it. So there's a portal protein here, which uses ATP to pack DNA inside. And this is how our multi-resolution DNA molecule came handy. So we basically use whatever we develop for origami to simulate packaging of DNA. So there's a portal, and we apply forces to DNA, and we packaged it. So that was 38,000 base pairs goes into the capsid. But then we go through this iterations of refinement, and at the end of the day, we arrive with a fully atomistic model of the packaged viral capsid. I think that's the first model of its kind ever built. And then we can run it using OLATOM microdynamic simulations and study all kind of things about it. So I'm not going to spend too much time. It's just kind of at this time, I just want to say that we can do it. What's interesting is that as we do the packaging, we can also measure the pressure inside of the DNA array. And the nice feature of our workflow is that pressure is preserved regardless of the resolution. So, we packaged it, the pressure increases, and we change the resolution, pressure doesn't change. Then we go to OLATOM, and we basically get the same pressure, which is kind of cool. Quantitatively, our numbers are in line with experimental observations very nicely. So here's the data from Ivilevich's lab. And everything we try to look at, we kind of reproduce what we know about the virus, a specific one, so we can compare the layering of the DNA and also the scattering profiles. So it all works very nice. Of course, we also know many other things that others don't know, like what exactly is the structure of DNA? How does it form contacts with the protein? Are there any defects in the packaging? How does the twist affect packaging? What's the ion atmosphere inside? So all of that is something that we are writing up right now. And I And I wish I could tell you more, but unfortunately I'm out of time. So at this point, I guess I'm gonna stop with some conclusions. The conclusions are kind of really simple. One main conclusion, I guess, is to say that, right now with the computers that we have and with the force fields that we have, we should be able to get numbers. Not just DNA does wiggles like that and something like that. No, I think we actually can. It's become a quantitative tool for predicting things, not just for making pictures. So that's kind of a big message, number one. Number two is I actually personally strongly believe in this multi-resolution modeling of systems, because with all atom, despite all of the progress in exascale computers coming about, and that us being able to simulate a small cell in all atom details probably in 5 years, that's always exciting and it's happening. Still exploring biological processes, with all of the chemistry and complexity, will require a multi-resolution approach where you basically go from coarse grain level to all atom, back to coarse grain, through all of the details. All of that's happening at the same time. So that's kind of an area where my lab is going with multiple projects, and well, maybe I'll tell you about it at some other time. And at this point, I'll just like to conclude and highlight people who did the work. So yeah, Jijung did most of the DNA work that I talked about, including parametrization and this DNA interactions and DNA loops. Chris did the multi-resolution DNA model, which was extremely important for us to make possible this viral packaging simulations, which Kush did. And David helped us with nucleosome simulations, and we've been enjoying a wonderful collaboration with Stagjip, Ha, and Hajin, and his student. So thank you very much, and I'm slightly over time, but I hope it's okay.
Thank you for the very nice talk. Are there questions or comments?
Can I ask a question? This is Debashish.
Sure.
Please.
This is regarding this last part. So in one of the capsids that I know, 529, which was studied by Bustamante, the force was about 60 piconewton. That is the force produced by the motor at the capsid entry. So this one seems to be weaker, or I'm looking at different quantity.
So there's two things. One is the force, and the other one, I mean, probably the stall force, and the other one is the pressure inside.
Yeah, that's why.
So the pressure, the pressure is slightly higher in the other virus. I mean, it's okay. We get 13, he got 16, but also the virus is slightly different. Unfortunately, for this particular virus, for HK97, there's no direct measurement of the pressure. So we could extrapolate what it should have been from other viruses, and it should be around like 13.
So other question I had, sorry, I didn't hear correct. That was 60 piconewton. So that was one of the strongest.
No, yeah, that's right. No, but that's a stall force. That's a force, so you have this packaging motor, and then you use a tweezer to ask the question, when does it stop packing, right?
Yeah.
So that's 60 piconewton, right? So in our simulations, we apply smaller force actually to package the DNA. I think it's about 10 piconewton or 20 or something like that.
Okay.
But of course, we have much less friction in our system. That's why the packaging actually proceeds faster than an experiment. There's always something, there's always, you have to cut corners, because that under experimental condition, packaging happens like in a couple of minutes. And even with coarse grain simulations, simulating something that takes a couple of minutes is challenging.
The other question is that there was a question of mechanism. Does it go helically inside, or does it go by some other? So when you are pushing it, so what is the mechanism through which it goes in?
So that is a really excellent question, and that's exactly what we set out to do. So what we did, we simulated packaging using 3 different ways. One is so-called straight packaging, when we don't have any kind of a helical twist to it. So we just, and that is actually enough to see all the nice toroidal layering of the DNA inside of the capsid. The other one was where we introduced a little bit of a twist informed by experiment.
Mm-hmm. So
the twist is actually small there. It's like 10 degrees per turn or something like that. So it's a really small turn. And of course, when we do the packaging, we don't do 1 simulation. I think we do like 10 simulations per each. And there we already can see some differences in the way DNA is arranged, but it's not dramatic. And then for the sake of it, we also did one where we like really twisted, like if you were to assume that the DNA were to do a corkscrew motion as it goes through the portal. So we also did that. In that case, you end up with quite a bit of plectoniums inside. And those are kind of arranged on the surface and then move about and dissipate. And, some of those, so we basically did 6 simulations we do many more coarse grain simulations, but all atom simulations are really expensive. And there we could pick only 6 systems, but still we run for a microsecond, each of them, 30 million atoms. Yeah, that was insane. But yeah, but it's I think it's the first simulation of that kind. Yeah.
Okay.
So basically, yes, we did this, but the effects are on global structure. There are noticeable effects on the global structure. We didn't see any effect on the pressure. We were actually expecting to see the pressure would differ if you kind of twist it or not, but we didn't see it.
Okay. Thank you.
Hi, Alek. This is Prabal here. Very nice talk.
Hi, Prabal. Thank you.
Yeah, so I have 2 questions. One regarding your Mr. DNA. So when you start from the very low resolution and grow eventually at different stage to the fine resolution, so what happens? I mean, here in your case, the example you showed, in many cases there are DNA secondary structure. They're forming loops, circles, correct, in later part. So how do you grow from the say very bead level 4 base like you are mentioning at some point that 1 bead equivalent to 5 base pair or 4 base pair, correct? That's what
right.
So how do you grow preserving all the secondary structure, the case where DNA do have different secondary structures?
So I mean, so first of all, within mRDNA, there are kind of only 2 possible secondary structures for the DNA. One is a double-stranded DNA, and the other one is single-stranded DNA. Okay.
Okay.
And that is kind of set at the beginning. So everything is operating with those 2 type of secondary structures. So we don't have quadrupluxes, we don't have I motifs and things like that, but those can be put in. That's not a problem. But the actual, so if you're asking about how we change the resolution, so that is done so we use spline to interpolate where the center of the helix is, and then we just resample the splines.
I see.
So that's actually easy part. For orientation, it's a little bit tricky because you also have to keep track where the minor groove and major groove is. So for that, we have orientational beads, but then we use quintillions and do interpolation in that space. So that also gives you continuous way to change resolution for the rotational degrees of freedom.
Mm-hmm.
So that's actually how we do at the technical level. And the tricky part is actually going from the finest coarse-grained model to all atom. Yeah. And there, yeah, so there we just know how to do it because we've done it so many times. So there's some transformations and things. So what's kind of nice about this mRDNA is that the structures that you get at the end is MD ready. You know, there are I mean, there are some other packages that can produce all atom structure from DNA origami design, but I don't know if you've tried to run them. Sometimes you can, sometimes you cannot. But ours is just runs. So that's one thing. If you're interested in like possibility of DNA helicing playing, like whether there could be a bubble formed, right? That's a very good question. Yeah.
I think, yeah, that's what I was.
Yeah. So in that case, we actually do not cover it in our model. What we do cover, we cover export to oxDNA. So our mRDNA package produces everything you need to run oxDNA on it. And as you know, oxDNA would actually, if something wants to break, it can, okay? So that is the route that we take when we suspect that there could be some distortion in the channel, in the structure. And then you can do two things. Once you have the oxDNA results, you can go back to mRDNA if you want, or you can go all atom simulation.
I see. The other question I had regarding this sequence-dependent flexibility, where you are showing that
Right.
the nucleosomal the protein, basically histone protein, when I mean, so depending, so do they sense? I probably did not follow there. I mean, when you have the flexibility, so and then when the histone DNA, histone proteins, do they have some preference, flexible part versus non-flexible? Is that, I mean, I did not follow quite there apart from the
Okay, yeah, sorry, I went very fast.
Apart from the accessibility, I was thinking within the mini circle, depending on the patches of highly flexible versus non-flexible part, is that something also you track or what?
So for nucleosome, so first of all, you know, when generally when we talk about, and by me, I mean me, like and like TechJibha, when he talks about flexibility, it's an operational definition. So it's for the whole fragment of the DNA.
Oh,
right?
I see.
Right. It's not like, I mean, usually that's what looping experiment measures, right? So when they measure, it's like the property of the sequence of this 90 base pairs. So what we try to do, we try to rationalize that global property, knowing the local stiffnesses. And in our model, we have local stiffnesses at the level of dinucleotide, dinucleotide approximation. So, I mean, we're not the first people to do it. Of course, it's, you know, there's lots of models that try to do the same. The big difference is, I mean, big or not, you can argue, but the difference is that we actually do it for deformed DNA. So, you know, our training set is curved DNA. Whereas like there are a few other models, they use the straight DNA. And the other one, I think the really cool thing is this inside-out transition. I believe this is kind of a new thing. Was somewhat overlooked. We also did this simulation that's in the paper. I don't have slides for it, but basically what we did, we took the 601 sequence, which is a well-studied nucleosome positioning sequence, and there's a crystal structure for them. And so that one, we can build 2 mini circles out of it. As you know, in nucleosome, you have like almost two spools of DNA around the nucleosome. It's a little bit less. And then we can take those two parts of the DNA as a mini circle, and we just run it using our calibration approach, and they will form exactly the right inside-out contacts, you know, just like what you find in a crystal structure. So that's kind of the relation between mini circles to nucleosomes. So I think our model describes mini circles extremely well, including this inside-out conformation. Now with the nucleosomes, so we did a few of them just to see whether we actually get the right inside-out contact, and we do.
Like whether attempt to
Yeah,
compute, say, persistent length in that form in mini circle and the difference between when they are not in a circular form?
So what we did, I mean, I really skipped over. I could have probably given a talk about that work just itself. So what we also did there, we tried to predict the looping rates, from we know the deformation free energy, and then we have to assume that there is a certain conformation that is at the barrier. Okay. So that is something that you have to assume. But once we assume that, we can compute what is the free energy cost of reaching that barrier. And that's basically how we end up with all those absolute values. And it worked pretty well. I mean, it's not perfect, but it worked pretty well for sequences that were published. And then we also designed a few sequences to have differential looping times. And that's what was validated by experiments. Now, with regard to persistent length, I think we probably will get it. I mean, it will be a sequence-dependent persistent length.
Yeah. But I'm curious.
I think
what will be the
right? I think it was roughly the same. I mean, the number you would get. I mean, I don't think you have, because usually those numbers are for like genomic length DNA, right?
Mm-hmm.
And I'm pretty sure that we just get that number from our stiffnesses. We just have to integrate them.
I see. No, because some years back we did this persistent length for the DNA wrapped around histone protein, and they found we came out to be very different than the normal.
Oh, cool.
Yeah, so that's why I was curious. And here you have a very long scale simulation to the sampling, and this inside out, all these are better sample than what we did. So that's why the calculation. Yeah, that will be very interesting to know.
Sure.
Okay, thanks.
Thank you.
Any other questions? I had a small question regarding that slide where you were showing the loop, and you were actually looking at the time series of two different distances.
Okay.
So my question was,
this one, right?
Anyway, you go to the slide actually. My question was, is it, yeah, exactly this one. Now, is it, I mean, have you measured what is, I was just curious, how does the distribution look like? This is the time series, but do they look Gaussian if I just look at the length distribution?
I think so. I mean, we didn't do we didn't plot it, but if you, I mean, the plot, if you look at the distance, it looks, yeah, I probably it is Gaussian. I would be surprised if it's not.
Okay.
Because why would you prefer, like, if you would have an asymmetric, then there should be some preference over it. I mean, what's interesting here, we didn't see any kinks. We would see kinks if we get like different linking number. Then we definitely see kinks. But for this one, it just stays.
But you mentioned that other groups are finding kinks. That's what you had mentioned.
Yeah, well, we also find kinks if we don't restrain. So the point is, if you make a mini circle, okay, and you run it unrestrained, chances that the mini circle is in that preferred conformation, poloidal conformations, are close to 0. So the DNA will rapidly try to find its local free energy minimum, and it will break. So what we do, we do not allow it to break immediately, starting from whatever artificial structure we do. So it can do this transition inside out. Once it finds its transition, then we let go, and it stays like that. And it can still do, all of these things. But that basically was the main difference. So if we don't do it, we would get kinks.
Okay.
Because the initial conformation is kind of far from equilibrium.
Okay. Any other questions or comments from anybody else? I don't see any. Then let's thank Aleksei once more for a fantastic talk. And
Well,
also
thank you very much for having me.
And also that brings us to the end of the conference. Your talk was the very last one. So
I know.
I would also like to take this opportunity to thank the organizers, Debashish, Prabal and Ambarish, all three of them, for putting together this very nice meeting. And even when the situation was really difficult. So thank you, everybody.
Thanks, Shakuntala. I'll now take this opportunity to give the final vote of thanks. First of all, I would like to thank all the speakers for putting together and giving excellent talks throughout the meetings. And of course, the session chairs for conducting the meeting efficiently on time and handling the question and answer session. And then finally, the participants who really, for last 2 weeks, very diligently tried attending most of the talks and raising interesting questions. And then, of course, the ICTS administration for hosting this meeting virtually and giving all the logistical supports. And then the ICTS program team is an excellent job. They really, right from the conception of the meeting to the arranging the talks over Zoom, everything was done so efficiently that it went glitch-free to the end. So I really thankful to all of the ICTS program office staff for doing an excellent job. And thanks all of you once again for being here.